Good day, everyone. This is Dr. Ruba Al-Husseini, teaching biology for AS students, Cambridge syllabus for 2025 examination. Hope my presentation is useful for you as you study, and let's begin. As you will notice with all my presentations, I would like always to begin by showing you the part of the syllabus which we are going to discuss. Here, please note that the part of shown here from numbers 1 almost to number 5 are practical skills, and these are going to be discussed separately in a different presentation, hopefully. It's a bit difficult to teach practical to those who are not face-to-face, but we will try our best. The rest of the topic is all about cells and their structure and the function of each part. So we need to know about the cells and the different types of cells, such as eukaryotic and prokaryotic, and the different structures that they may contain and what does each do. So briefly, just have a look quickly on the different parts, which we're going to discuss in full detail as much as we can. First of all, we're going to talk about the light microscope. It is actually the way by which we discovered this lovely and beautiful world of cells that cannot be seen by a regular eye. So first of all, this was an invention that made us discover many worlds that were not easy for us to see. Let's begin by talking about that the light microscope is called light microscope because it uses light. Light comes from the mirror. As you can see at the bottom of the microscope, it is reflected by the mirror from the surroundings, and the light passes through a group of lenses that help to condense the light to make a most amount of light reach the specimen that you will obviously place on the stage. This area in the middle is called the stage, and then you're going to focus onto this specimen using a number of different objective lens. This is what we call them, objective lens. And then the light travels, as we all know, in a straight line up to the very top end of the microscope. That is what we call the eyepiece. This is where your eye will land to see the specimen. So this is a way by which we can make use of the light and a group of lenses to magnify our specimen and to be able to view it. Light waves have an average wavelength of, bear in mind, you may have studied in physics that light is actually 7 different colors. So because the light around us is a combination of all these 7 colors, so that's why we refer to the wavelength of light as an average wavelength, because as we use each light color differently, we will get a different wavelength. So the average wavelength of light is 400 nanometers, and this is a very important number to remember. You might put it in your notes to help and memorize it. Also an important number is the maximum magnification of a light microscope. Light microscope, the maximum, some books say it's 1,000 times, some books say it's 1,500. Well, it must be according to the type of microscope that you're using. So this is another important number to remember. Your eye's resolution is around 0.1 millimeter, while that of a light microscope is 0.2 micrometers, or to put it in a different unit, 200 nanometers. So we will talk about resolution in more detail just as soon as we start comparing the light and the electron microscope. So be patient with me a little. But we all have a knowledge about resolution, especially when we're talking about screens and how they're much developed than before, and how the The image produced by the new cameras have better resolution, so you might have a fair understanding of what this means. A live specimen could be viewed with none or simple preparation methods. That's the best thing about light microscopes. You can view something as it moves, as it lives. If it has something that looks like a heartbeat, you might be able to see the beats and count it actually under the microscope. So that's one of the advantages of the light microscopes. And also that specimens can be seen in color, which helps to better understanding of it as well. It is cheap if compared with the electron microscope, and it is easy to carry around. So that is all what we have to know for now about the light microscope. Moving on to the electron microscope. Obviously, this is something that is much more sophisticated, much more expensive. You will never, well, according to your school, but I haven't seen a school that has an electron microscope, maybe a university, of course, or a research center. So this is an electron microscope. It uses, instead of light, it uses electron beams to view the specimen. Electrons are focused using electromagnetic or electrostatic lenses. So they're not actually lenses, they're just equipment that help to move using electromagnetism the beams of electrons. So this is how electron beams are focused. Electron beams have a wavelength of, let's pause a little bit here, it's 0.005 nanometers. Do you remember the previous light microscope? It was 400 nanometers. So can you note the great difference between this size of wavelength and the light microscope? Well, how does that help us? It helps in many ways, and that's why I told you let's wait and talk about resolution in a minute. Wavelength affects the resolution of a microscope. So more is going to be said about resolution in a minute. Maximum magnification here, wow, that's 10 million times of magnification. Again, this is according to the type of microscope. You might find different numbers in different textbooks. So this is a number that is a bit different from a textbook to another. Your eye's resolution is 0.1 millimeter, but the resolution of an electron microscope is 0.1 nanometer. Wow, much smaller, a much more precise resolution than our eyes, which, well, obviously, that's why we made this piece of equipment. It helps us to see the things much better. Now here there is a limitation. Only dead specimens could be viewed. The specimen goes through many steps to be viewed, and one of them is removing water from the sample, dehydrating it, staining it with special stains that is mostly made of heavy metals that will obstruct the electron beam and work like the stains that we use in light microscopes, so that it interferes with how light passes through it, which makes things look darker or lighter in contrast to one another. So these very drastic processes through which the specimen goes through kills it. It's dead. It cannot survive without water. It cannot survive with much heavy metals inside of that cell. So all of these steps just causes the death of the specimen, and that's a limitation. Also, one other thing, that images that are produced are only black and white. We cannot see in color. Color can be added through computer generated wizardry, if we would like to call that. This is what we can do with by artificially coloring it using computer programs. It is very expensive. Yes, it is. We're talking millions of whatever Anyways, much larger than the light microscope, and it must be kept in a room with controlled temperature and pressure. That's why you don't find it easily. Now, here we're going to talk about magnification and resolution and the differences between these two words. Magnification refers to how many times, the number of times a specimen is enlarged. This tells us how many times it is enlarged. Here we have two cells. You can notice here that the nucleus, if these were the same cells, I'm not sure if they are the same cells or not, here is a nucleus and it's much larger. It looks like a white blood cell to me actually. So that is normal in white blood cells. They do have larger nuclei. So the magnification here is not obvious. These are two images produced from two different microscopes. But here you can note that the only structures visible in the first cell over here, it is the cell membrane, the cytoplasm, and the nucleus. These are the only three structures visible in the first cell. Underneath the other cell that is white and black, we can see that we have more structures that are visible. So here we can find a sausage-shaped object, and this is the mitochondrion. Near it, a group of stacked membranes, which we will learn is called the Golgi apparatus. And over here, this is the nucleus, and in the middle is the nucleolus. So here we can see many different objects and structures inside the cell, not only the cell membrane, cytoplasm, and nucleus. So here we have a greater resolution, because resolution helps us to see in more detail, helps us to see more structures. And resolution has a definition of the ability to distinguish between 2 points, 2 close points. So if I have 2 close points, here we have a different way of showing things. These are 2 images from 2 different microscopes, but they are of the same sample, same tissue. These are plant cells. On the right-hand side, we can see the same cells as on the left-hand side, but there is a difference. You can notice that with your own eyes, can't we? Well, they have different resolution. They have the same magnification, by the way. Same magnification. They're both magnified. I don't know how many times. I did not capture that from the book, but they have different resolution. And the one with better resolution is the one on the right side. Okay? And why do we say that? Because, well, we can see more clearly. Things are not blurry as they are seen in the left photo. So we can see chloroplasts, and actually I can see starch grains inside these chloroplasts. Here we have a nucleus, and this big structure filling up most of the cell. This is the vacuole, as we can see it over here as well. But here we can see in more clear detail. So that's why this one on the right side is the electron microscope image, and this one is the light microscope image on the left hand side. Easy? Let's move on. Here we explain the difference, or actually the relationship between the wavelength and resolution. Because you remember, I told you to remember the numbers. Light microscope has an average wavelength of 400 nanometers, and this is, if you have studied physics, you would know that any 2 crests in a wave, a distance between them, or 2 troughs, it doesn't matter which way you take the measurement, but any 2 close crests to one another, we can measure the distance between them, and that is called the wavelength. And here light has an average wavelength of 400 nanometers. What is the significance of that number? Well, we can see that these wavelengths are stopped by certain objects. Here it is a mitochondrion that has a diameter of 1,000 nanometers, which is obviously bigger than 400 nanometers. So they will stop these light beams, and in the end, your eye will receive light from areas, but this certain area, which was the mitochondrion, less light will pass through it. So it appears darker, so you can see it. But if we have smaller structures, and yes, we do have much smaller structures in a cell, such as ribosomes, that have a diameter of 25 nanometers, much smaller than 400 nanometers. And because of their small size, they do not stop light waves. Light waves can pass through them just easy. They don't stop light waves. So you cannot see anything different at the other end of the microscope, which tells us why we cannot see clearly some stuff. All right? Here, if I had an electron beam, and this is a very poor diagram that I drew with my own hands to show that we have smaller wavelengths, all right? It should be even smaller than that, but to help you understand, these will be blocked by these small structures such as ribosomes. So the electron beam, because it has a much smaller wavelength, will be stopped by these smaller objects, so they are visible on the other end of the microscope. So that's why we can see it, and that's why we have a better resolution using an electron microscope. Right, so here are some pointers to remind us of the units that will keep coming up as we discuss samples under the microscope. So let's remember and let's memorize actually the following. A meter contains 100 centimeters. Yes, we all know that. Of course you do. And each centimeter is made up of 10 millimeters. So a meter contains 1,000 millimeters. Now, if we chop up a millimeter into 1,000 pieces, we get 1,000 smaller units that we're going to call micrometers. So a millimeter is made up of 1,000 micrometers. Moving on, a micrometer can be chopped into smaller pieces. Yes, we need to do that because we have very small structures inside cells, so we have to make up a new unit. Nanometer is even smaller than a micrometer, so 1 micrometer contains 1,000 nanometers. You should be able to interchange from one unit to another. This is a very important skill. Many questions on your exam paper will test you on the ability of changing the unit. All right? So 1 millimeter is actually 1 million micrometers, and this is another way of writing it down. And 1 millimeter, so if it's 1 meter, it is 1,000,000 micrometers. And again, 1 meter is 1,000 million nanometers, 10 to the power 9 nanometers. All right, hope this is easy to understand. Now, we're going to go into the organelles. And organelles actually comes from the word organ. We know what an organ is. An organ is a part in an organism that has a certain function. But we use this word in a way, making it a smaller. Organelle is a smaller word for organ. So these are structures found inside cells that have a specific role and function. And we are going to divide the organelles that we're going to see into 3 different categories. One of them is being not surrounded or not bound by any membranes, such as ribosomes, centrioles, and some other structures. I can't remember much, but these are 2 examples. Another example of organelles, but they do have a membrane surrounding them, just one membrane, so a single membrane bound organelle could be a lysosome, and we'll know what lysosomes are in a minute, Golgi apparatus, and vacuoles. And there are more structures. These are just examples. You can tell us more as we move through the syllabus. Finally, we do have also organelles that are surrounded by a double membrane, nucleus, mitochondria, chloroplasts. And I like giving an example, making this easier to understand or easier to remember. Mitochondria are the power stations that produce energy. They are very important parts of a cell. So maybe we need a double membrane to just ensure uh, that they are protected. Maybe when we learn more about mitochondria, maybe we'll understand the reason why there is a need for two membranes. But again, here we have a nucleus which holds the very precious molecule DNA that holds information that without it a cell cannot function. So why not protect that important structure with a double membrane and leaving small spots through which molecules can enter or exit a nucleus, as if we have checkpoints to ensure that nothing damaging enters into the nucleus or nothing important escapes the nucleus. I would say that's a good way to remember which organelles are double membrane bound, such as a nucleus, mitochondria, and chloroplast, and which have a single membrane and which have none. All right, it's according to their importance, of course. Let's move on. So here we're going to talk about prokaryotes. And prokaryotes, I like to remind everyone, it starts with this pro. Pro, let's remember prototype. Yes, the word prototype helps us because prototypes are the first invention that you made. It's the prototype, and then you were going to make it better and better and better. So prokaryotes are let's continue and talk about what karyote means. Karyote is nucleus. So these are cells that are primitive cells. They have no nucleus in the sense of what we know is a nucleus. We just talked about the nucleus having a double membrane. So these prokaryotes have no true nucleus. As you can see here, the DNA is a circular DNA that is lying free in the cytoplasm. Where's the double membrane? Where did it go? Well, it doesn't have any. It's a primitive cell. Well, okay, so that's why we gave it a certain name and put it in a certain group. So all bacteria are prokaryotes. They do not have a nucleus as we know the regular nucleus, and the DNA is just lying in a space inside the cytoplasm that we called a nucleoid. This area is a nucleoid, right? Or a nucleic acid, the DNA, you can call it this way as well. Other structures that are known to be found in a bacterial cell, well, they do have a cell surface membrane like all other cells. They do have a cell wall. Cell walls, but they are made of a certain chemical. They are made of murine, a type of a chemical called peptidoglycans. Beneath that cell wall, yes, we know there is a plasma membrane or a cell surface membrane, cell membrane, whichever you call it, is all correct. And the cell membrane is similar to that of eukaryotic cells. Some bacterial cells have, let's just wait a bit, because I would like to start by discussing the parts of the bacteria that are found in all bacterial cells. This is a typical bacterial cell will have the circular DNA, will have cytoplasm, will have cell membrane, and will have a cell wall. And of course, ribosomes. Do we have ribosomes? Yes, here. These are ribosomes, and they have a special size. We call it 70S. So these are the basic things that you would find in any bacterial cell. Let's revise. DNA, circular DNA lying in the cytoplasm, of course, ribosomes also lying in the cytoplasm, and then a cell membrane and a cell wall made of murine. Other structures in this diagram are extra structures. You might or might not find it in a bacterial cell. So let's go over them from where? Let's start with the flagella. Flagella are long whip-like structures, and they keep beating in the solution in which the bacteria is living, so helping it to move. A bacteria could have one flagellum or many flagella, one thing. Bacterial cell could be surrounded by a capsule or a slimy layer that helps to keep the cell from drying out if it's in a dry condition, or helps it to protect it from antibiotics attacking the cell. All right. Other things, well, sometimes we find enfoldings in the cell membrane. Some of them have the ability of making photosynthesis. Some of them, like the mesosome, work in a way similar to that of mitochondria, by which we mean it releases energy or produces ATP. Okay, which way you say it is correct. Plasmids are small DNA circles that are found in the cytoplasm. So these are also optional. You might or find them or you might not find them. Finally, the pili are small structures that help to attach two cells together and through which information can be shared. Plasmids can move from one cell to another through pili. Did we miss anything? Yes. Well, we can go to the next page. Here, we kept talking, but we said everything over here except for plasmids are actually a way of exchanging information between cells, bacterial cells, and sometimes they help each other by gaining antibiotic resistance. So a bacterial cell has information that helps it to stop the antibiotic from causing harm to it, and it can share this information through sending a plasmid to another bacterial cell. So aren't they small but bright? They have a way. Anyway, bacteria feed by extracellular digestion. They are saprophytic organisms. They release enzymes to the surroundings, and the enzymes digest the surrounding cells. If they are maybe carbohydrates, they will be changed into sugars, and then these small molecules, because of their size and because they are soluble, can be absorbed by the bacterial cell and thus take its food. This is how they feed. Glycogen granules and lipid droplets are found inside the cytoplasm of bacteria. They are good for food storage. Here are what we have said earlier about ribosomes. They are the factories that produce proteins. They are the ones that are responsible for making proteins. Ribosomes are found in all cells, in all living cells. So bacteria have small type of ribosomes called 70S. Eukaryotes, we will learn in a minute that they have bigger ribosomes and we call them 80S. Respiration helps in a certain enfolding called the mesosome. We talked about the flagella. We talked about bacterial cells transferring plasmids from one cell to another, and that's what biologists consider as a primitive way of transferring DNA. So this is conjugation in a primitive way. Now let's move to the eukaryotes. Eukaryotes are animal cells, plant cells, not bacterial cells, animal cells, plant cells, fungal cells, and finally, what else? Protist cells. So the other four groups of organisms that do not belong to prokaryotes, eukaryotes. So we're going to start with animal cells as an example. And here you can take some time to look at the structure, the basic structure of an animal cell. You can find all these, and we're going to talk about each in detail. Plasma membrane, or cell surface membrane, or cell membrane, is the exterior wall, or let's not call it a wall, it's a flexible substance but it is a boundary, a very intelligent boundary that helps to control what goes in and out of the cell. Between here we have two cells, each of them has its own cell membrane, and in the middle we have a space that we call intercellular space. So here chemicals can be moving from that space into a cell, or the other way around if a cell needs to get rid of something. So a plasma or a cell surface membrane is a boundary between the cell and its environment. It can control the movement of materials of the cell, very intelligent to have the ability to obstruct something that might hurt a cell. Good, so I can stay alive by preventing things that should not enter my cell. If it's too much water that will cause my cell to burst, then I should be able to stop it. If maybe too much salt or too much or a chemical such as a heavy metal, if there's a way to stop it, that would be great. Now here we need to note the size of the plasma membrane, 7 nanometers, and I also suggest that you memorize that number. So many numbers here, we would like to memorize it to be able to compare sizes in an animal cell or a plant cell. More will be said about the plasma membrane in another chapter, so that's why we said very little over here. A nucleus, that's the main part of the cell that holds the very important information. The DNA is here inside this largest organelle inside the cell. It's surrounded by a nuclear envelope. Now remember how many membranes surround the nucleus? Who remembers? I hope you remember there are 2 membranes, and in the middle you might find certain spots that we will call pores. We'll talk about that right now. They contain pores that allow the movement of molecules into and out of the nucleus. The DNA is concentrated in this place. It is wound up, or wound up is the best word that we can use to describe how the DNA is not a straight thread, because it will take such a big space if it is. So if it is not being used, the information inside the DNA is not being used, then it will be densely wound up around itself, and it will have a dark color. But if it is actually these spaces, or it might look like a space, but this is a lighter color because the chromatin over here, the chromatin is just another word for the DNA. So the chromatin might appear as light in color or dark in color. And that is just because either the DNA is dense because it is wound up around itself, or it is stretched to be able to be read, or making copies from it, or whatever. So that's why we have chromatin that fills up the nucleus, that has certain areas that are lighter in color. We call the lighter part the euchromatin. It starts with an EU and then chromatin. Or the dense part are called heterochromatin. Okay? The nucleolus, and this is the part highlighted here, this is a darkly stained part. This is still the DNA, still containing DNA, but this DNA produces ribosomes. So it has a certain role of producing ribosomes. And ribosomes will not stay inside the nucleus, they will work outside. So they have to be a small size to be able to pass through our, yes, this is the line showing us the nucleolus. Over here are the small spots that we're going to call pores, and obviously this is the nuclear envelope surrounding the cell, the nucleus. So let's continue. Now we're going to talk about a structure that is found just as we move out of the nucleus. In actuality, this structure is continuous with the double membrane that surrounds the nucleus. The nuclear envelope over here, as you can see, is made of 2 layers, and this layer is extending into a group of latent sacs, a system of sacs that are interconnected with one another. We call this structure endoplasmic reticulum. Now you will note that this endoplasmic reticulum has on its outer surface many ribosomes. And these many ribosomes give it the appearance, as you can see here, these are the dots over here. These are the ribosomes on the outer surface of that endoplasmic reticulum, which made it appear as a rough structure to those who saw it and called it the rough endoplasmic reticulum due to the presence of these ribosomes, many ribosomes. Now, if you remember, we just said that ribosomes are the parts of the cell that are responsible for protein synthesis. So if they are closely found next to the rough endoplasmic reticulum, well, maybe they do something together. Yes, they do, as the ribosome makes the molecule, the protein molecule, because that's what ribosomes make. This protein molecule is threaded inside these sacs, that rough endoplasmic reticulum sacs. So this is where the proteins are kept. They are kept inside the rough endoplasmic reticulum and they can move. Remember, there were channels. You might not be able to see the channels here because this is a one plane, but there are channels connecting these sacs to one another. So this helps to transport proteins in a way that it is not affected by whatever chemicals are found in the cytoplasm, right? So any cell that produces a lot of proteins must have an extensive rough endoplasmic reticulum, right? So any cells that produce a lot of proteins, such as a cell that is responsible for producing digestive enzymes in our digestive system, will have that many cells, that many structures of rough endoplasmic reticulum. Let's move on. Here we see a similar structure, but this is not rough. Yes, it has no ribosomes on its surface, and this has a different name and a different role. This is called the smooth endoplasmic reticulum. They are again a group of latent interconnected sacs, but they're different. They're not continuous with the nuclear envelope as that of the rough endoplasmic reticulum. The function of the smooth endoplasmic reticulum, and one thing that we can say about both endoplasmic reticula is that you can use RER as an abbreviation for the rough endoplasmic reticulum and SER for smooth endoplasmic reticulum. You can write it in your exam paper. That's totally fine. It's totally accepted, just like writing RBC to refer to a red blood cell. Also, note that you have to use capital letters when you do that. All right? So it is the site of lipid production. All right, so this is where lipids are produced inside the cell. Steroids are chemicals that are lipid in structure, and especially some of our hormones, especially female sex and male sex hormones such as estrogen, progesterone, testosterone, all of these are steroids. So these are produced in the smooth endoplasmic reticulum, and they contain detoxifying enzymes, so it's another thing that they can do. They're found in cells that produce, oh, I think this is wrong. We should remove this part. They should be found in cells that are rich in, or their responsibility is to produce lipids or hormones, steroid hormones, okay? So this is not correct. Ribosomes, they're very small structures. Remember, they are 20 to 25 nanometers in size. Well, that is according to which organism we're talking about. Smaller ribosomes are found, remember where? In prokaryotes, yes, you're right. larger ribosomes, 25 nanometer in size, in eukaryotic cells. Okay, they are made of two subunits. One is larger than the other. Over here, we have this big subunit, and another one beneath it. It's smaller in size. It's a bit smaller. They are mostly attached to the rough endoplasmic reticulum, but they all also occur freely in the cytoplasm. And if it's a bacterial cell, well, it doesn't have any rough endoplasmic reticulum, so they will be found freely in the cytoplasm. They are made from ribosomal RNA. More about RNA and DNA will be mentioned in a separate chapter, but just let us know that the chemical that makes a ribosome is ribosomal RNA and proteins. They are involved, obviously, as said before, in protein synthesis. Now, the Golgi body, or another word, Golgi apparatus. Both are correct. This is a tightly packed group of latent sacs, and these sacs have different sizes. You can see that the sacs are increasing in size as we go down, and then they decrease once more. That's normal in the shape of a Golgi body. We have two faces. One face is close to that of the rough and smooth endoplasmic reticulum, and is actually the face that receives vesicles that are budding off from the rough and the smooth endoplasmic reticulum. So carrying chemicals that should not be mixing with the cytoplasm, and then it fuses with that of the Golgi apparatus. Well, what would this Golgi apparatus do? Well, it has also a role of synthesis of proteins, but usually it works more in modification. So it was produced, the protein, but we still have to add things to it. Maybe change it into a glycoprotein. So we're going to add a carbohydrate to it, maybe. Also modification of lipids and modification of carbohydrates. And in the end, this chemical that has been modified is packaged. You can see here it is budding out of the last layer in the Golgi apparatus, and this vesicle will separate, and then it will move. We have another diagram showing that nicely. So here this is a vesicle coming from the endoplasmic reticulum. It fuses with the Golgi apparatus, and then it is modified, and then it buds off, and then it is shipped until it reaches the cell membrane of that cell, and then it fuses with the cell membrane, thus releasing the chemicals outside to the exterior of a cell. So this might be a chemical that the cell wants to secrete. Maybe it's a protein, enzyme, maybe it's an antibody, who knows? It's a chemical that is to be shipped outside of the cell. All right. We do have other structures that also have one membrane around it. Now, rough endoplasmic reticulum and smooth endoplasmic reticulum both had one layer of membrane surrounding the interior, and this is also a lysosome. It's a small vesicle between 0.2 to 0.5 micrometers in diameter. It contains a mixture of enzymes, but these are not just any enzymes. These are lytic enzymes. Lysis means breaking things into smaller parts, and lytic enzymes are enzymes that break things into smaller parts. And this is helpful if a cell has an old organelle that is not functioning well. So lysosomes are released onto that part of the cell to digest that old organelle. Lysosomes can be shipped outside of the cell to also kill something or get rid of something digested. So lysosomes, obviously, these are chemicals that will destroy anything inside or outside a cell, so they have to be surrounded by a membrane to prevent them leaking out into the cell and causing harm. So they have to be under some certain control. So they contain enzymes that will digest something inside or outside the cell. We have an example here. This is a white blood cell, a phagocyte that performs phagocytosis. You must have learned about that in your O-level biology. And here is a substance. It could be a food particle, or it could be a bacterial cell or a virus that has been engulfed and taken into that white blood cell. And the white blood cell now has taken in that chemical or that, let's say, foreign body, to make it easy. And then these lysosomes will fuse with the vacuole in which that substance is. And that will break it down into smaller pieces. Well, if it's food, then the food, or if any chemicals are useful inside of that, it will be useful, absorbed into the cell and made use of. But if these are harmful chemicals or have no use for the cell, then that vacuole will fuse again with the cell membrane, and these chemicals will be released or exocytosed or removed by exocytic processes. Lysosomes have many jobs. Let's look at the jobs. They supply the enzymes that are needed for destruction of old or extra organelles. They digest material taken into the cell, as we said in white blood cells, maybe taking in the foreign microorganisms and trying to digest them, the process called phagocytosis. They also destroy whole cells and tissues, and that is normal. Many examples, such as we have the muscles of the uterus after giving birth. There is no need to keep holding onto these muscles. There is no child inside the uterus anymore. So they are broken down by these lysosomes. The milk producing tissue after a child has stopped breastfeeding, so there is no need for the tissues that produce milk anymore. So that's also, these structures are broken down by lysosomes. Mitochondria, a very important part of the cell. It is the part that releases energy from food by aerobic respiration. It makes a very important molecule that we call ATP. ATP molecules are what we call the energy currency inside the cell. This energy currency molecule will be used elsewhere inside the cell to drive many different chemical reactions and processes. So without a mitochondria, a cell, well, I feel for it. It's not going to survive. Mitochondria have a double membrane. The first one is, the outer one is smooth. The second one, the inner membrane, has many folds that we call crista. Okay, this is how we say it, which provides a large surface area for chemical reactions that are going to take place inside the mitochondria. There is a liquid-like structure that fills the mitochondria. We have a name for it. It's called the matrix. Ribosomes are found inside this matrix, and a loop of DNA is also found in the matrix. DNA is usually inside the nucleus. What brings the DNA here? Well, that also made scientists think. And, well, they found that the presence of the DNA and ribosomes will help the mitochondria synthesize its own proteins. Well, that's useful because this cell is going to make energy, or this organelle is going to make energy for the cell. So if it's able to make its own proteins, its own enzymes, that would be useful. Yeah. All right? Any cell that is very active, such as muscle cells, cells like those that have active transport occurring in great amounts, such as neuron cells, nerve cells, they would have large numbers of mitochondria. Again, the liver cells, they have a lot of chemical reactions happening in them, and a lot of ATP is needed for that to happen. So many mitochondria will also be found in liver cells. Microtubules, another way of saying it, microtubule organizing centers. Microtubules are something that are extraordinary because they are actually roads inside a cell. It's a road where molecules can move from one place to another. They also help to give structure to the cell. So together, all these tubules help to give the cell structure, especially if it's a cell that has no cell wall to support it. So it makes up the cytoskeleton of that cell. So microtubules are long, rigid, hollow tubes found in the cytoplasm, and they are made together with other filaments. They make up the cytoskeleton. The cytoskeleton helps to determine the cell shape. So that would make sense now if you think about the red blood cell, and we have always referred to red blood cells in O-level biology as having a biconcave shape. How does it keep its shape if it doesn't have a cell wall that is rigid to keep that shape? Wouldn't that be a valid question? What helps that cell to keep its shape is the cytoskeleton inside of it, right? Microtubules are made from a protein called tubulin. And tubulin, here, this is tubulin, is actually made of two parts together, alpha tubulin and beta tubulin. These two molecules combine together to make a dimer, and the dimers together make that structure. Actually, if we look at the dimers, and here we're looking from one side, on the other side behind it is the other dimer, tubulin. Here it should be orange in color, alpha tubulin. So here we can see how they are arranged, and they are actually 13 dimers together. Have a look. So this is number 1, 2, 3, 4, 5, as we go until number 13. So these are the 13 dimer molecules that together make one ring, and this rings are held together, one after joined together end to end, making us the protofilament. The protofilaments line up to make this ring form with a hollow center, and now we have a microtubule. What do they have? What role do they have? Microtubules are mechanical function for support, as we said, gives the cell its shape. Secretory vesicles and other organelles can be moved from one area to another on the outer surface of the tubule. So yes, it is actually a road, and there's a lovely video. I hope I added it to the last presentation slide, I hope, of how the structures are moved along this road, I would like to call it. Anyway, let's move on. So secretory vesicles and other organelles can be moved from one. This is brilliant, because I have been reading many years ago that chloroplasts have the ability to move from one side of a cell, obviously a photosynthetic cell such as a palisade cell, from one side to another according to how much light is that cell receiving from each side. And how do they move? We have never seen flagella, for example, on chloroplasts. So how are they moving? Well, they must have been using these roads. Yes, they must have. So this is a way of a transport system inside the cell, these microtubules. Membrane-bound organelles are held in place by the cytoskeleton. So yes, sometimes we find a structure such as a nucleus in a certain spot. It does not move from that spot, so maybe that is why it is held in place using the cytoskeleton. Now we're going to talk about another structure, and this structure is only found in animal cells, so it's not found in plant cells. looking at it using the electron microscope, these are actually two cylinders, two hollow cylinders, but they are lying at 90 degrees to one another. So if you're looking in this image, we can see one that looks like a ring because we're looking at this face, and one that looks like two lines. Well, because we're looking from the side, so you can only see two lines of those. So this is called the centriole, and we still have no full understanding of what centrioles do, but we find them, as we said, lying in pairs, lying at 90 degrees to one another, near the nucleus of, again, animal cells. It's not found in plant cells, in an area that we're going to call a centrosome. The centriole is made of 9 triplets of microtubules. So each of these straw-like structures, we're going to call it a microtubule, and we have 3 of those together, and we have 9 of these triplets making up that centriole. Okay, very, very small structures and very, very brilliant. Now, scientists think that these help the movement of chromosomes as the cell division begins, because, as you know, half of the chromosomes move to one pole of a cell and the other half move to the other side, the opposite pole. So they think that they help with the movement of the chromosomes. Basal bodies that are found in cilia, we're going to talk about cilia in a minute, act as microtubule organizing centers. The microtubules extend from the basal bodies into the cilia and flagella, and these are essentially for the beating movement that happen. Let's move on. Now, cilia and flagella. These are slender, microscopic structures, especially cilia. They're much smaller than flagella, and they extend from the surface of many cells, such as ciliated cells, for example, that line our respiratory tract. The length of a single cilium, one of them is called the cilium, it is between 1 to 10 micrometers. Width is about 1 micrometer in diameter. Cilia are broadly divided into 2 types. So some of them are motile, they move, and this is what we find in the lungs, respiratory tract, the middle ear, and actually also in the ovary, the oviducts, all right, helping the egg to move from the ovary to the uterus. So these have rhythmic beating motion, and this will help to clear the airways from the mucus that holds the dirt and bacteria. This will help us to breathe easily, and helps, if it's a flagellum, it will help to propel cells such as here we have protist cell, I hope, or if we're talking about sperm, it's the same thing. Sometimes the cilia are non-motile. They look like appendages, like microtubules on the surface of cells. They can bend with urine in the kidney to give a signal that there is urine flowing. Okay, so that's this role. Flagella are similar to cilia, but they're much longer, usually found in fewer numbers on one end or opposite ends of a cell. We find it in prokaryotes, obviously, and sperm cells, some cells that help them to move by the whipping action that they do. Microvilli are little microscopic projections. Here we can see them on the surface of a villus. Here we have microvilli on the surface of a villus, and they help to increase the surface area even more, so that chemicals such as enzymes break the food down and increase the surface area for absorption of the chemicals, the food that has been broken down. Mostly found in the small intestine, on the surface of egg cells, as well as white blood cells. So they help to increase the surface area and maybe attach to foreign bodies if we're looking at white blood cells. Thousands of microvilli form the structure called the brush border. that is found on some epithelial cells, such as in the small intestine. Generally, they are shorter and smaller in diameter than cilia, so in comparison to cilia, they're much smaller and shorter. They are commonly about open. Well, you don't have to learn all of that. Just remember that cilia are bigger than microvilli and flagella are bigger than both of them. All right? But let's continue. Microvilli are essentially bundles of cross-linked actin fibers. Although they are cellular extensions, they are little or no cellular organelles present in microvilli. However, they are covered with their own plasma membrane, so this is one thing that we can know. It is covered with plasma membrane, and it contains some cytoplasm and some microfilaments. Don't bother yourself much with that, but just let's move on. Now, the plant structures. Now, these are structures that were not discussed before because we can only find them in plants. Of course, the most important part is the cell wall, and because it is too important, there is a special chapter for the cell wall. So more will be discussed further in another lesson or another chapter. Now, the cellulose cell wall surrounds all plant cells. It is made of the main structure or the main chemical that makes up the cell wall is cellulose, and these are fibers that are cemented together. The cell wall is composed of many layers. The middle lamella is the part that holds adjacent cells together. So that's a layer that is between cells, and here you can see that the thin black line between these two cells, this is what we call the middle lamella. Plasmodesmata are actually spaces or channels, if you would like to say, between two cells. They help to transport chemicals and water and whatever is dissolved in water from one cell to another. So this helps from the cell-to-cell transport. That's good because now we have a thicker wall around the cell, which will make it a difficult process for chemicals to move through that thick wall. So that's a good way to find a way to move substances from one cell to another. Pits are another thing that is a space also, but here we needed to start by saying that a cell has a primary cell wall, it is on the outside of the cell, and a secondary cell wall as the cell gets older in size, older in age. Okay? It builds another wall that we call the secondary wall. So if we want to help movement of substances from one cell to another, let's not make the walls too thick. So let's have small gap-like spaces, and this is what we call a pit. So it's an opening in the secondary wall of one cell, and there's another one similar to it in the other cell. So these help also to transport substances, mainly water, from one cell to another. Now, cell walls have many functions. They provide rigidity and strength to the cell. That's why the cells do not burst if it has a lot of water inside of it. And this helps to give the cells strength by being turgid, by being filled with water. Unlike animal cells, they don't have that option. They will burst if they have too much water inside of them. It helps to force the cell to grow in a certain way, and they give the cell its certain shape. So some cells are rectangular in shape, some of them are angular, some of them are circular. It all has to do with the shape of the cell wall that gives that cell its shape. The cell wall can be reinforced with extracellulose, so we can add more cellulose to make that cell more thick or more strong, and it will be thick actually, or we might add a different substance. So there could be extracellulose or there could be another substance. And lignin not only gives strength, it will make the cell impermeable to water. So water cannot pass. If it has lignin to pass through it, it will not be able to pass through the areas that contain lignin inside a cell. But it gives a lot of strength, and that you will find in the woody parts of a plant. Cell walls are freely permeable. That is very difficult to imagine, because you think of a walls in our rooms, as impermeable. But let us think of cellulose in a different cellulosic fibers. Actually, here look like bars, as if you have a container made of bars of maybe a certain metal, maybe iron bars. But the iron bars have spaces between them. So chemicals smaller ones only, can pass through these bars. So that's why it is fully permeable. But if lignin or another chemical that we will learn later called suberin is lining that cell wall, then it will become impermeable. Okay? Plant cells are connected to nearby plant cells by thin strands of cytoplasm. This is what we called plasmodesmata. If we were referring to one of them, it's called a plasmodesma, which pass through pore-like structures in the cell wall. Movement of substances through the pore is thought to be controlled by the shape of the pore or the structure of the pore. So it's not everything can pass through these pores. So it's a way of controlling what goes in and out of the cell again. Let's move on now. We're talking about the plant ultrastructure again, and we're talking about a different structure, the vacuole, also called the cell sap vacuole. Okay. In animal cells, we have small temporary vacuoles. Yes, we will find small things, but they're not there all the time. These are just like vesicles. They move around. They send something from one part of the cell to another. So as we can see in phagocytosis, there will be a vacuole in which the substance engulfed is situated. Now in plants, we don't have the same thing. We have a large central, permanently filled with a fluid that we're going to call the cell sap. So the cell sap is a liquid-like substance that fills up the interior of that vacuole. The cell sap is a mixture of sugars, salts, pigments. Here we have a lovely microscopical diagram, and obviously because we can see color, so this is using a light microscope. These are the cells, and can you see inside the vacuole? We can see the vacuole because of the pigment stored inside of it, and these are the cells that contain that purple pigment that helps this lovely flower to have its lovely color. All right? So many things can be stored inside of that vacuole together. Sugars can be found with salts and pigments and waste products, along with water, obviously. All of that surrounded by a membrane that we're going to call a tonoplast. And again, just like the cell membrane, it can control the exchange between whatever is inside the vacuole and the exterior, the cytoplasm. Okay, so it's the same as the cell membrane. Functions of vacuoles: they absorb water by osmosis, pushing the cytoplasm away, so making the cell turgid. It stores food, such as sugars and mineral salts. It stores pigments, just as we mentioned in a minute earlier, to give plants their color, such as in petals. Sometimes they accumulate wastes that are produced by metabolism. They are byproducts of metabolism, or sometimes toxic chemicals, or sometimes unpleasant tasting chemicals, so that whatever is eating that plant will be discouraged to continue because it doesn't taste nice. All right, let's move on. Plant, again, ultrastructure. Here we're going to talk about chloroplasts. Remember, it shares something with mitochondria. It has a double membrane. These green organelles are surrounded by a double membrane containing a fluid. Yes, mitochondria used to have a fluid. We used to call it the matrix. Here it's not called the matrix. It has a different name. We're going to call it stroma. Here enzymes carry out some of the photosynthetic reactions. We have a stack of structures called thylakoids. They are stacked one over the other, and together a whole stack has a certain name. We call it a granum, and these contain the chlorophyll. So thylakoids contain the chlorophyll, which will absorb the light energy to produce ATP. All right, so we can produce actually food, and then that food is turned into ATP. And this is a very short way to say it, but chloroplasts produce sugars, and then the sugars are turned into carbohydrates, and then that could be digested into sugars and then broken down inside the mitochondria to release energy in the form of ATP. So we here made a very big jump. The stroma also contains free ribosomes and starch grains, and just like the mitochondria, a circular DNA is found inside the chloroplast again. So there are many similarities between chloroplasts and mitochondria. They both have a double membrane bound envelope, if you want to say it. They are organelles that are double membrane bound. They have a fluid in the center. They both contain DNA, circular DNA, and they both have ribosomes inside of them. So now we have 4 similarities, if you want to say, between mitochondria and chloroplasts. Now we're at the final plant ultrastructure that we find in plants, amyloplasts. We don't find them in all cells. Just like the chloroplasts are found in some cells. So amyloplasts are found in cells that are working in storage. So such as potato cells in the potato tuber, or in other tubers such as sweet potato and so forth. These organelles have the function of storing starch, and here we can see many of them, and they are blue in color, or you might refer to them as violet if you want, but this blue color is because we added iodine solution. Iodine solution reacts with the starch to produce that lovely color. Obviously this is an electron microscope. No, sorry. This is a light microscope image because we can see in color. All right. So we find these inside the amyloplasts are found where starch is stored, usually in roots or tubers such as potato and sweet potato. They have a double membrane. They contain both types of starch, and we're going to learn about starch in the next chapter. There are 2 types of starch. One is called amylose and amylopectin, so both of them are found in amyloplasts. Some of them show rings, concentric rings. I'm not sure if you can see them over here in this image, but here you can see them in this diagram. Now we come to viruses. Viruses are actually very tricky things. We cannot call them cells. They don't have the cellular structure. But what defines a cellular structure? We have just studied many cells, bacterial cells, animal cells, plant cells. What do they all have in common? They all have a cell membrane, and they all contain a liquid called cytoplasm, jelly-like substance called cytoplasm. And this is not found in viruses. Viruses don't have a cellular structure. Also, viruses don't have that ability, or the 7 characteristics that you might have studied in the O level, that are specific to all living things. All living things can move, respire, are sensitive, they have the ability to grow, They reproduce. We said respire, so reproduce, and they excrete, and they show nutrition. They can do all 7 things, but viruses don't do all 7 things. Actually, they do only one, and in certain condition, if they are inside another living cell, they actually hijack that cell. I feel like they are like pirates. They enter inside the living cell and they control everything inside of it, actually causing harm to that cell. So that's a problem. Viruses are always parasites. They invade cells, causing them harm. This is how they make copies of themselves. They reproduce using whatever is inside a living cell. They make that cell make copies of that virus. Okay, so viruses actually are considered by scientists being on the borderline between living and non-living things, and that is because of these reasons that we gave. Okay, and they have a smaller size than all other things that we have discussed earlier. They're smaller than eukaryotes. They are much smaller even than prokaryotes. They range in size between 20 to 300 nanometers. Remember, 20 nanometers is almost as equal to a ribosome. See how small they are compared to a eukaryote. All viruses are parasitic, which means they can only reproduce by infecting and taking over living cells, as we just said, causing them damage. The DNA or RNA in the virus uses the host cell's resources to make new copies of itself. So the basic structure of a virus is that it has a nucleic acid. That nucleic acid could either be a DNA strand or an RNA. So it's either this or that. Some viruses have DNA, some viruses have RNA. And then surrounding that DNA or RNA is a protein coat. We call this a capsid. Okay, the protein coat has another name called capsid. And this capsid is made of smaller molecules, smaller units that come together to make the capsid, and this we call capsomeres. So this is the basic structure of a virus. Some viruses have an envelope that is made from actually the host cell that it invaded. So it's taking a blanket, a part of that cell membrane of that cell of the host, as if it is surrounding itself. Maybe it's hiding itself, might be. Very tricky things. We cannot call them cells. We're going to call them particles. Okay, so they are not surrounded by a partially permeable membrane. They do not have a cell structure. As we said before, they do not contain cytoplasm. So that's in regards with viruses. This is the final part of our presentation. Here we're talking about the organization of cells. Cells cells, if they are in multicellular organisms, they will be arranged in a way so that they help each other. So cells together make tissues. So tissue contains either of one or, well, we're AS students now, so we have a little bit more space in what we call a tissue. So here we have a tissue. You can see that this is ciliated cells in conjunction or working together with goblet cells that produce the mucus, and we still call it a tissue. So it is one or a few amount of cells, different cells, that work together to perform a certain function. We have epidermal tissue, we have muscle tissue, we have many different types of tissues. Organs are made up of various tissues grouped to work together and perform their function efficiently. So a leaf is an example of an organ in a plant, and the heart or the eye is another example of an organ in a human being, if we're talking about eyes or animals. Systems comprise of various organs that work together to perform large-scale functions, such as the reproductive system, maybe the digestive system. Many people believe that there are no systems in plants. That is one way of thinking about it. The digestive system in humans is an example, as we said. Now we have reached the end of our presentation. I hope that it was not difficult. I hope it was easy to understand. I hope it was interesting to you. You can reach out through the messages if you have any questions. Here I have added, I hope you can some way or another get to see these different YouTube videos that are very interesting. To me, they are very interesting. And one of them is going to talk about the endosymbiosis theory that I did not have enough time to talk about. But maybe you can find out about it. It's very important to know. So that's the end of our lesson. I hope you enjoyed it. I hope it made you the studying of this chapter easier. To see you in other lessons. Goodbye.